Hello everyone, welcome to Unacademy. Uh, I'm Debashree here. Today my topic of discussion is your recombinant DNA technology, which is very important, right? So if you want to join Unacademy Plus, you can join it with the help of my referral code DEBA1. This is my referral code. With the help of this referral code, you can join Unacademy to get the 10% off. What are the benefits of Unacademy Plus? Our daily live classes, structured courses as per the CSIR syllabus, live tests and quizzes, unlimited access to all the educators at single subscription. If you want to join Unacademy Plus, you can join it with the help of my referral code, that is DEBA1, to get a 10% off. So let's move to the price list. So if you join for 1 month, the price will be 1890. For 3 months, it is 4725. For 6 months, it is 7560. For 12 months, it is 9450. And for 24 months, it is 11340. If you want to join Unacademy Plus for any for 1 month, for 3 months, you can use, and you can also use my referral code, that is DEBA1, to get the 10% off, right? So let's recombinant DNA technology. So if we define recombinant DNA technology, recombinant, if you see the name, it is basically recombination of DNA. That means, suppose this is one organism, this is another organism. This is one organism. Let me change the color for your better understanding. This is one organism, and suppose this is another organism. When this combines, and suppose I have these are the DNAs of the two different organisms. This is one organism, this is another organism, right? So when I combined these two, I'll be getting a new type of DNA, right? And that is what I named it as third type of DNA, which is basically a combination of first and second. Which is what? It is a combination of first and second. So that means this DNA is a combination. Combination of what? Recombination of first and second. So we call it what? This is what we call it recombinant DNA technology. It is basically a set of techniques that enables DNA from different sources to be identified, isolated and recombined, so that new characteristics can be introduced into organisms, right? So this is basically we can say that this is a techniques which can be used to isolate, identify and recombined recombine, so that a new character can be added to an organism, right? So this technology basically used to produce you can say a hybrid organism with an additional character, right? The DNA cloning was engineered by Stanley Norman Cohen and Herbert Boyer in 1973. So in 1973, these two scientists basically worked on this, and the name of the scientist was were Stanley Norman Cohen and Herbert Boyer. The Paul Berg discovered first recombinant molecule, that is the DNA from simian virus 40 and lambda phages. So Paul Berg is the first scientist who what he has done, who produced the recombinant DNA technology by the combination of SV40 virus and lambda phage. Right? So what we have seen in this slide is recombinant DNA technology is basically a techniques that enables the DNA from different sources to be identified, isolated and recombined, so that a new characteristics can be introduced into an organism. Right? So the DNA cloning was engineered by Stanley Norman Cohen and Herbert Boyer in 1973. The first recombinant product, or you can say recombinant molecule, was produced by a scientist called Paul Berg. He basically produced a recombinant DNA from a virus, simian virus 40 and lambda phages. Right? So now let's see the DNA cloning. If we talk about the DNA cloning is what? It is a production of large number of identical DNA molecules from a single ancestral DNA. That means suppose this is a ship, right? If we produce a cloning, there is no any kind of crossing over, or there is no any kind of, you can say, variations, right? The shape will be exact similar. Suppose this is a ship 1, and if I make the cloning of the ship 1, say I have done the cloning of the ship 1 and I got another ship which I named it ship 2. But since this is a cloning, that means the ship 2 will be identical, will be very much identical to ship 1. So we can say DNA cloning is basically the production of large number of identical DNA molecules from a single ancestral DNA molecule, right? It is basically two types, cell-based and cell-free. This cloning can be either cell-based or cell-free. We will see this. Cell-based. So if we talk about cell-based, it is the first form of DNA cloning to be developed. The DNA cloning first developed was cell-based. So we can call it, it is a first form of DNA cloning to be developed. In vivo cloning method. This is in vivo. In vivo means what? Inside the living organism. Inside the living organism. In vivo stands for inside the living organism, right? So this is an in vivo cloning method, right? Cell mediated. Obviously, if it is taking place inside the body of the living organism, then that means this is cell mediated. It is taking place inside the cell, within the cell, right? So we call it cell mediated. And as it is taking place inside the living organism, we can call this living organism as host, right? So we can also call it as a host mediated, or you can say that this process requires a host, right? Okay. Now coming to cell free. Cell free is basically in vitro. In vitro means what? Within lab, in lab, in laboratory. We call it what? In vitro. In vitro means that is not done in, you can say in vitro means what? Which is not done inside the living organism. It is something, you can say, outside the cell. It is not cell mediated, right? So we call it in vitro, means it is taking place inside the lab, right? So we call it in lab. This is enzyme mediated process. That means enzymes are used to carry out the process. It is a PCR. PCR. You know what is PCR? PCR is your polymerase chain reaction. Polymerase chain reaction. It is used to produce it is used for amplification of DNA, right? Produce many copies of DNA. So this cell-free is basically what? It is a in vitro process, means the DNA cloning is done inside the lab, not inside the living organism. So this is basically carried out with the help of some enzymes and with the help of some instruments. One of the example is your PCR, right? Okay. Now let's move to the tools for DNA technology. If we check the tools for the recombinant DNA technology, the first is the enzyme for DNA modification. The first tool for recombinant DNA technology is your enzymes, right? So what are the enzymes? DNA polymerase, nuclease, ligases, and end modification system. These are the enzymes which are used for the recombinant DNA technology. DNA polymerase, right? Nucleases, and modification system, and ligases. We will discuss each of this. Coming to DNA polymerase is an enzyme that synthesizes new polynucleotides complementary to existing DNA or RNA. Let me explain you in this diagram how DNA polymerase works. If this is a DNA, say this is 3' to 5' DNA, right? So what will happen? A complementary, and this is say 5' to 3', right? And we all know that synthesis always takes place from 5' to 3'. So what is happening? Say here, synthesis takes place from 5' to 3'. So what will happen if this strand is getting replicated? Replicated means what? Getting copied, if this strand is getting copied, that means what? A new strand will be synthesized. The new strand will be synthesized with the help of deoxynucleotide, polymerases and ligases, right? So the enzyme DNA polymerase, what it will do, it will add the DNA polymerase will add nucleotides here, right? So it enzyme synthesize. So we can say that DNA polymerase enzyme synthesizes new polynucleotides complementary to existing DNA. Suppose this is, let me tell you here, it is A, T, G, C, right? So if this strand is getting copied, that means if this strand is getting copied, that means what? The complementary strand will be synthesized, right? So for making the complementary strand, what will happen? DNA polymerase will come, and what it will do? It will make A, T, A, C, G, right? So this addition of nucleotide is done with the help of DNA polymerase, right? DNA polymerase is what? It is DNA dependent DNA polymerase, and DNA pol-1 is also known as Kornberg enzyme. Since it was discovered by scientist Kornberg, so it is also known as Kornberg enzymes. RNA-dependent DNA polymerase is known as reverse transcriptase. So if we talk about DNA polymerase, there are mainly DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. So many types of DNA polymerases are there, but DNA polymerase 3 is most important for application, right? And DNA polymerase 1 is known as Kornberg enzyme because this was discovered by the scientist Kornberg. There is another polymerase enzyme which we call RNA dependent DNA polymerase. That means how RNA dependent. So that this RNA dependent DNA polymerase, what it will do, suppose this is what? RNA, right? Suppose this is RNA. So this enzyme will synthesize DNA from the mRNA. So what it will do? This enzyme, this RNA dependent enzyme helps in the synthesis of DNA from mRNA, right? As it is producing DNA from mRNA or from RNA, so we called it RNA dependent DNA polymerase. His name is reverse transcriptase. Coming to DNA polymerase, I told you different types of DNA polymerases. So if we check the DNA polymerase, you can see here the types of DNA polymerases. This is DNA polymerase 1, tag DNA polymerases, and this is reverse transcriptase. First, we will see the DNA polymerase 1, which is also known as Kornberg enzyme, right? So what are the functions of Kornberg enzyme is it has got 5' to 3' polymerase. 5' to 3' polymerase means what? It adds nucleotide in the direction 5' to 3'. So we call it 5' to 3' polymerase. And remember one thing, synthesis always takes place from 5' to 3', right? So we call it 5' to 3' polymerase. That means addition of nucleotide. It helps in the addition of nucleotide. Addition of nucleotide, right? Coming to 5' to 3' exonuclease. 5' to 3', if it is 5' to 3' polymerase, then we call it addition of nucleotide. But if it is 5' to 3' exonuclease, then it will stand for primer removal. That means primer gets removed for 5' to 3'. 3' to 5' exonuclease stands for proofreading. 3' to 5' is your proof reading. Say synthesis is taking place from 5' to 3', right? 5' to 3', what is taking place? Synthesis, right? Synthesis 5' to 3'. Primer removal also 5' to 3' exonuclease. But proofreading will be ulta. It will be 3' to 5' will stand for your proof reading, right? So that is what is the DNA polymerase 1. This is the property. These are the properties of the DNA polymerase 1. Coming to tag DNA polymerase, if we say tag DNA polymerase, it is isolated from the bacteria Thermus aquaticus. What is Thermus aquaticus? Any idea, any one of you? Thermus aquaticus is a very heat stable bacteria. That means what? Heat stable bacteria means what? It can withstand a very high temperature. Thermus aquaticus means what? This bacteria This bacteria can withstand very high. This bacteria can withstand very high temperature. So we call it thermus aquaticus, right? And its activity is 5' to 3' polymerase, also 5' to 3' exonuclease. So 5' to 3' polymerase. Polymerase means what? Addition of nucleotide. And 5' to 3' exonuclease. Exonuclease means what? Removal of primer, or you can say primer removal, exonuclease, right? So that is all about the Taq DNA polymerase. Next we will see reverse transcriptase. So if we talk about the reverse transcriptase, what is reverse transcriptase? I already told you it is basically the synthesis of DNA from RNA, right? It is primer dependent. Because why it is primer dependent? Because suppose this is mRNA and this is DNA, right? This you can say RNA and this you can say DNA. So now here DNA is produced from RNA. So which enzyme will be used? With the help of enzyme RT. RT stands for your reverse transcriptase, right? So now what will happen? This enzyme is a primer dependent. Primer dependent. Why primer dependent? Because primer provides 3' hydroxy N for addition of the nucleotide, right? So reverse transcriptase is primer dependent. Without primer, it cannot carry out the process, right? So we can say that reverse transcriptase is your primer dependent enzyme because primer provides 3' hydroxy N for the addition of 5' phosphate, for the addition of DNTPs with 5' phosphate, right? So that is why it is primer dependent. It is RNA dependent, and this reverse transcriptase enzyme was discovered by Howard Temin and David Baltimore. It also works for 5' to 3' polymerase. That means addition of nucleotide, right? So that is all about your enzymes, DNA polymerase. Next, move to nucleases. If we talk about nucleases, nucleases means what? It degrades DNA molecules by breaking the phosphodiester bond. So nucleases are the enzymes which actually breaks the DNA molecule. How it breaks the DNA molecule? It breaks the DNA molecule. How it breaks the DNA molecule? By digesting, or you can say by breaking the phosphodiester bond, right? So two types of exonucleases, two types of nucleases are there. One is your exonuclease, another is your endonuclease. If we talk about exo and endonuclease, exo stands for outside, right? So if I draw this DNA, exonuclease will be what? The enzyme which will act at this sites outside the DNA, means at the terminal sites, at the terminal sites, right? So that is what we call exonuclease. It removes nucleotide at a movement from the end of the DNA molecule. So it starts removing the nucleotide from the end of the DNA molecule, and it is known as exonuclease. But if we talk about endonuclease, endonuclease will work on this side. That means it breaks internal phosphodiester bond. It will work within the DNA segment, right? That's why we call it endonuclease, because it will work within the DNA segment, right? Within the DNA molecule. Okay. Some example of nucleases are RNase H. RNase H. H stands for hybrid. Here H stands for hybrid, right? Now, what is as in the name of the enzyme, you can see the word hybrid. So what it will recognize? It will recognize the DNA RNA hybrid. Because the name itself has hybrid, so it will recognize DNA RNA hybrid and cleave only RNA molecule. Though it recognizes both DNA RNA hybrid, but it will cleave or it will cut only the RNA molecule, not DNA, right? Coming to RNase A, if we talk about RNase A, you can see this cleave only single-stranded RNA, not DNA. RNase A will cut or it cleaves only your single-stranded RNA. It do not cleaves DNA, right? What it will generate, it generates a 5' hydroxy and 3' phosphate N. So once RNase A cleave what? Single-stranded RNA, it generates a 5' hydroxy and 3' phosphate N. Normally, see, this is a very unique property. Why unique property? Because normally there is a 5' phosphate and 3' hydroxy, right? But this is the only enzyme. Let me highlight it. This is the only enzyme which is producing your 5' hydroxy and 3' phosphate, which is producing your 5' hydroxy and 3' phosphate. So this is a unique enzyme. Normally what we can see is 5' phosphate, 3' hydroxy, or 3' hydroxy, right? But this is the case in the case of RNase A. We see that it leads to the production of 5' hydroxy and 3' phosphate. It breaks phosphodiester bond, right? Next, mung bean nucleases. Mung bean nucleases cleave single-stranded DNA or RNA, right? So mung bean nucleases are the enzymes which cut, or you can say which cleave single-stranded DNA or RNA, right? DNA is one. DNA. So name itself says DNA means what? It will cleave DNA. Both single-stranded as well as double-stranded. S1 nucleases is extracted from Aspergillus oryzae. Aspergillus oryzae, I think everyone knows, it is a fungus. It is a fungi, right? And it cleaves strand opposite to nick on the complementary strand. So it S1 nucleases cleave the single-stranded strand and also the nick strand. Suppose if I show you the DNA here, just a moment. Yeah. If I draw this DNA and say, for example, here there is a, just a minute. If this is a DNA and this is a nick, right? So S1 endonuclease can act at this nick site also. Nick, see this nick site. It can act on the nick site also. So this is how it will act on the nick site, right? So that is all example of some nucleases, right? Next, we will see the AND modification system. If we talk about the AND modification system, in the AND modification system, we have put certain, you can say we have put certain enzymes, like for example, alkaline phosphatases, T4 polynucleotide kinase, terminal deoxynucleotidyl transferase, right? So these are basically what N modification system. Alkaline phosphatase, what is the function of alkaline phosphatase? It breaks the phosphodiester bond and generates 3' hydroxy N and remove the 5' phosphate. And remove what? 5' phosphate. So alkaline phosphate, what it will do? It removes 5' phosphate and generates only 3' hydroxy N. Polynucleotide kinase is basically isolated from E. coli infected with T4 phages, right? It is isolated, or you can say it this enzyme. This is kinase. Kinase means what? Addition of phosphate. The function will be addition of phosphate. So whenever you will get the word kinase, make sure that this actually stands for addition of phosphate, right? So here if you see T4 polynucleotidyl kinase, it is isolated from the E. coli infected with T4 phages. It adds phosphate to 5' N. It helps in N labeling of the DNA molecule. So this polynucleotide kinase will add 5' phosphate, right? Also helps in the labeling of DNA molecule. Coming to terminal deoxynucleotidyl transferase. If we talk about terminal deoxynucleotidyl transferase, it usually adds DNTPs at the terminal site. It adds DNTPs only at the 3' end. It is template independent, primer dependent. This is also responsible for homopolymer tailing. Right, so that is what is about your N modification system. We have seen that N modification system basically deals with 3 types of enzymes: alkaline phosphatase, polynucleotidil kinase, and then your terminal dinucleotidil polynucleotidil kinase terminal dinucleotidil transferase. Right, so if we talk about alkaline phosphatase, these are the enzymes which breaks the phosphodiester bond and generates a 3' hydroxy and removes the 5' phosphate N. T4 nucleotidyl kinase is isolated from the E. coli infected with T4 phase. This basically adds phosphate group in the 5' N. Terminal deoxynucleotidyl transferase adds DNTP. It adds DNTP at 3' and this is template independent and primer dependent, right? Next, moving to restrictions enzymes. If we talk about the restriction enzymes, these were discovered by Werner Erber, Daniel Nathans, and Hamilton Smith. So these are the 3 scientists who worked on restriction enzymes, right? The scientists were Werner Erber, Daniel Nathans, and Hamilton Smith, right? So these 3 scientists worked on the restriction enzymes. Bacterial enzymes, these are restriction enzymes. What we called it? We called it is a immune system, or you can say defense system of prokaryotic organism, right? It is a defense system of prokaryotic organism, right? And what? Bacterial enzymes. That's why we call it bacterial enzymes, because these act as a defense mechanism for bacteria. Bacteria protect its own DNA by adding a methyl group, right? It cuts double-stranded DNA by cleaving the phosphodiester bond. So it adds, you can say, it cut double-stranded DNA by breaking the phosphodiester bond. It restricts the entry of foreign DNA in the bacterial cell. It targets double-stranded DNA. Restriction enzyme can digest, or it digests double-stranded DNA, right? It cut both the strands in sequence specific. It also cleaves both the strands in a sequence specific way. Right, so what we have seen in restriction enzyme, 3 scientists basically are involved in the development of restriction enzymes. These are Werner Erber, Daniel Nathan, and Hamilton Smith. These basically are defense systems of prokaryotes. So bacterial enzymes cut double-stranded DNA by cleaving the phosphodiester bond. It restricts the entry of foreign DNA molecule, and it can cleave or cut both the strands. Coming to your restriction modification system, there are, if we talk about restriction modification, just a minute. If we talk about restriction modification system, if we talk about restriction modification system, so restriction enzymes are of 3 types. What are the 3 types of enzymes? See here. Type 1, type 2, and type 3, right? So type 1 is homodimer, this is heterodimer, and this is again homodimer, right? Here it is what? This is ATP dependent, this is ATP independent, and this is ATP dependent, right? It needs a magnesium ion, it also needs a magnesium ion, it also needs a magnesium ion. right? Next, what? Here it is bifunctional. That means the enzyme act both as a recognition and modification system. Recognition, it functions both in recognition and modification. But here it is not bifunctional, it is unifunctional. Perform single function at the time. You can say unifunctional. This can be either, this will be either help in recognition or modification. This is also again bifunctional, right? Next, clear everyone till here? Next, here the cutting site is 1000 base pair upstream or downstream from cutting site is either 1000 base pairs upstream or downstream from the recognition site. So here cutting site is equals to recognition site. Here cutting site will be 25 base pair away from the recognition site, right? So these are the all restriction modification system. So what we have seen today, we have covered up the DNA recombinant technology. Here we have covered here recombinant DNA technology. So what we have seen in this is, we have seen that this is one organism DNA and say this is another DNA, right? So recombinant means what? This will form and give you can see this is a combination of two, right? So we called it recombinant DNA, right? To produce the recombinant DNA, we need many factors, many tools for the recombinant DNA technologies. First, we will see the enzymes. What are the enzymes we needed for this recombinant DNA? The enzyme we needed are number one, DNA polymerases, right? Nucleases, ligases, DNA polymerases, nucleases, ligases, and modification system, right? And modification system. And modification system basically deals with alkaline phosphatase, then T4 polynucleotide kinase, T4 polynucleotide kinase, and terminal transferase, dinucleotidyl terminal transferase, right? And this DNA polymerase is basically three types: DNA pol 1, DNA pol 2, DNA pol 3, right? It is also some other are tag polymerase and RNA dependent DNA polymerase, RNA dependent DNA pol, right? What we have seen, the function DNA polymerase 1, basically it has got 5' to 3' polymerase, 5' to 3' exonuclease, and 3' to 5' exonuclease, right? DNA polymerase 2, it helps DNA polymerase 1. DNA polymerase 3 is a main replicating enzymes, right? And here if you see tag DNA polymerase is obtained from the bacteria. It is obtained from bacteria Thermus aquaticus, which is very heat stable. This is what? This is a heat stable bacteria, right? That is all about your DNA polymerases. Next, we have seen the nucleases. Nuclease is basically DNase 1. DNase 1, what it will do? It will cleave both double-stranded as well as single-stranded DNA, right? Coming to ligases, what is the function of ligases? It helps in the attachment. The 3' hydroxy attacks 5' ligases are having a if we see ligases, suppose this is a ligase, right? Here at the lysine there is a presence of 3' hydroxy which will attack the 5' hydroxide of DNTP, of DMP, dinucleotide monophosphate, right? Then we will see this about ligases, we will be discussing it soon, right? Because it is not this is a summary I am drawing here, so it is not possible, it is not better to conclude it in the summary class. We will see this in the next class. For the time being, you should know that it helps in what? It helps in your attachment, right? And modification system, alkaline phosphatase. What will alkaline phosphatase do? It generates 3' hydroxy and removes 5' phosphate, right? Next, T4 polynucleotide kinase. T4 polynucleotide kinase, what it will do? It adds 5' phosphate. Dinucleotide terminal transferase. Dinucleotide terminal transferase, what it will do? It will add DNTPs at 3' end, right? So this is the entire, you can say, summary of the class. Summary of the class, right? So if you like it, please hit the like button. You can also subscribe to the channel, and you can also join the Telegram channel with the heading Let's Crack CSIR-UGC NET, right? So if you have any queries, students, you can comment it, and you can also write, you can also comment it, and you can also drop your queries and doubts. If you have any doubts regarding subscription also, you can comment, right? Thank you so much, students, for watching this sessions. Hope to meet you soon in our next class. Thank you.